In this video I will perform a plant DNA extraction followed by a PCR or polymerase chain reaction. You probably heard this term before related to COVID-19 testing, but here I use it for an application in plant biotechnology. I previously obtained mutant Arabidopsis plants with gene knockouts in a specific gene, meaning the gene is not functional anymore. That's why it's called a knockout. But this plant has 2 copies of the gene, and to investigate the function of the gene, I would like to work with homozygous knockout mutants, meaning that both of the copies of the gene are not functional anymore. So it is important to check homozygosity by PCR. To perform a PCR, we first need isolated DNA. Thus, the first step is a DNA extraction. I started by adding one small leaf which contains more than enough DNA and 400 microliters of Edward extraction buffer to a 1.5 ml tube. I grinded the leaf with a small pestle to obtain a homogeneous solution. This partially breaks the cells and exposes the insides of the cells, including the DNA, to the extraction buffer. To separate cell materials from the DNA and proteins in the solution, I centrifuged the tube at 12,000 RPM for 4 minutes. In the meantime, by the way, we use CRISPR to program these leaf cells to kill themselves. Cool! The green pellet you see here contains cell stuff like the cell walls, and the green solution above it contains our DNA. After transferring 300 microliters of the supernatant containing our DNA to a fresh tube, I added 300 microliters of isopropanol which will precipitate the DNA out of the extraction solution. To now separate DNA from the solution, another centrifugation was performed for 5 minutes. Now the little pellet you see at the bottom of the tube contains all the DNA, so the supernatant above it is discarded. This centrifugation step is repeated with 70% ethanol to remove all impurities from the pellet. Here I'm resuspending the washed pellet in 30 microliters of water. The DNA is now dissolved in the water. Our DNA solution is finally ready to be used for PCR. To identify the homozygosity of the plant, I had to do 2 separate PCRs actually. I already prepared 2 master mixes, which are all the ingredients the PCR needs to work. The first PCR reaction that will be used is performed with a forward and reverse primer that binds our gene of interest. The second PCR will use the same forward primer, but this time in combination with a primer that binds the mutant T-DNA that is only present in the mutated dysfunctional gene copies. The primers bind the DNA as shown here, generating a different band pattern for each zygosity. Thus, to figure out this band pattern, I did a simple gel electrophoresis after the PCR reactions were complete. Our little plant turned out to be homozygous because we had 2 short bands in the gel. Thanks for watching and I see you next time.